Traditional drug discovery has heavily utilized animal testing for predicting efficacy and safety of molecules during drug development. LifeNet Health is an organization that's been in business for over 40 years. And recently we built out, about the past 7 years, the LifeSciences division, where we're recovering tissues for research purposes, processing those tissues, isolating cells, and building in vitro models to help biotech and biopharma companies evaluate their compounds in preclinical drug discovery. And we're developing model systems that replicate the human condition so that we can develop more safe and effective drugs.
So the motivation for developing the MPS plate was really based on the current models and inventions for MPS that were coming out from other groups. This plate was designed with a biology forward thinking. The innovative models at LifeNet Health that we can use in the system are pretty amazing, actually. We've developed a liver model, for example, that is much more human-like, much more in vivo-like than any model that's currently available. And that means that it has the capacity to do transporter uptake and transporter movement into the bile. It has metabolism. It has longevity and culture. And these things, when you add them to an MPS platform, give that platform even more power in predicting toxicology and DMPK parameters.
The MPS plate works by tying together multiple various organs of one's choosing through a semi-permeable membrane. We have two versions, one that allows for recirculation and the other that's a linear flow system. And this allows for a lot better control over the system and the test. So this MPS system has its advantages in that it's a larger scale than most of the commercially available systems. It allows for one to take many kinetic samples over a long period of time without disrupting the test. It also allows for one to take many different endpoint assays, depending on what one is looking for. I believe the most exciting aspect of this is furthering the in vitro and vivo equivalency in order to somehow and someday replace animal testing. It allows us to create a model that is hopefully far more relevant to human physiology and human biology, while still helping to do our due diligence with toxicity and screening.
The MPS system is only as good as the cells that go into it. And we take an approach of using human primary cells, where traditionally in drug discovery, they're using immortalized cell lines, which do not show the same function and characterization as human primary cells. With today's technologies, an MPS offers an opportunity to improve that process by having an in vitro system that better models in vivo, human in vivo model systems.
TruVivo is a novel cell culture technology that we developed to meet a lot of our internal needs, where it has extended features or properties that allow us to do studies for longer while maintaining the properties of primary hepatocytes for much longer, especially as they relate to toxicity testing, metabolism, and applications like that. So TruVivo is revolutionary, or at least provides some additional values and benefits compared to traditional models or other current platforms that are out there, and that it's much simpler and easier to use while retaining these key functions and being stable over time that typically are only achieved using more complicated, complex systems. Our scientific collaborators, and even with our own research groups, are finding all kinds of new ways to use TruVivo, because now it opens the door to doing experimentation and new study designs that, frankly, we couldn't do prior to having a system like this.
Through LifeNet Health and the gift of organ donation, we have access to a lot of biospecimens that are unique and very well characterized that not a lot of other CROs have access to.
So one of the big motivators for me, and I've been in the in vitro testing space for 25 years, has really been the ability to start replacing animals, to start reducing animal usage, to start testing drugs in a human model to predict toxicity and movement in a human, which really makes the most sense to me. We're trying to predict safety in humans.